Good morning, everyone. I'm Fabi George. I'm doing my MSc in Biotechnology at Kristu Jayanti College, Bengaluru. My roll number is 19LS301010. So I'm here to talk about northern bloating and western bloating, two of the different types of bloating techniques. So for the introduction, what is bloating? Bloats are nothing but techniques for transferring DNA, RNA, and proteins onto a carrier so that they can be separated and often follows the use of a gel electrophoresis. So there are different types of bloating, like mainly there are three types of bloating: southern bloating, western bloating, and northern bloating. So southern bloating, it was used to detect DNA. Southern bloating is the oldest type of bloating found, and it was developed in the year 1975 by Sir Edwin Southern. Northern bloating is used to detect RNA, and western bloating they are used to detect protein. So let's come into our topic, that is first northern bloating. So northern bloating, it's a technique used for the detection of specific RNA sequences. Northern bloating was developed by James Alvin and George Stack at Stanford University at 1979 and was named such by analogy to the southern bloating. RNA was separated by size and detected on a membrane using hybridization probe with a base sequence complementary to all or a part of the sequence of the target mRNA. Now let's see the process of northern bloating. So the first step involved in the northern bloating is RNA is isolated from several biological samples. It can be various tissues or various developmental stages of the same tissue, etc. We all know that RNA is more susceptible to degradation than DNA. So the next step is samples are loaded onto the gel and the RNA samples get separated according to their size on the agarose gel, and the resulting gel following after the electrophoresis run. Then as we can see in this picture, the gel is then gets blotted on a nylon membrane or nitrocellulose filter. How? By paper, by creating the sandwich arrangement. The next step on northern blotting is that the membrane is placed in a dish which contains hybridization buffer with a labeled probe. So it will hybridize to the RNA on the blot that corresponds to the sequence of interest. The membrane is then washed to remove the unbound probe. And the last step is the labeled probe will be detected either via autoradiography or via chemiluminescence. Chemiluminescence is used only when a chemically labeled probe will be taken. Only then we can use chemiluminescence. So either if we use chemiluminescence or autoradiography, this both of them results in the formation of a dark band on an X-ray film. And now the expression patterns of the sequence of interest in the different samples, they can be compared. Now let's talk some applications of northern blotting. They are helpful to study RNA degradation, study RNA splicing, RNA half-life, etc. Then detection of mRNA transcript size. This technique is often used to confirm and check transgenic mice or animals. Some of the advantages of this technique, that is northern blotting, is that it is used to detect RNA size. Then the quality and quantity of RNA can be measured on the gel prior to the blotting. Membrane can be stored and reported for years after blotting. So there are also some disadvantages for this northern blotting, like it's a time-consuming procedure. RNA samples may get degraded by RNases. Like if RNA samples are even slightly degraded by RNases, the quality of the data and quantification of expression is quite negatively affected. So that is a major disadvantage. So that's a small video on northern bloating. So now let's come into our second topic, that is western bloating. So the Western blot, sometimes also called as protein immunoblot, is a widely used analytical technique in a molecular biology and immunogenetics. It is used to detect specific proteins in a sample of tissue, homogenate or extract. So Western blot is often used in research to separate and identify proteins. In this technique, a mixture of proteins is separated based on their molecular weight and thus by type through gel electrophoresis. And these results are then transferred to a membrane, producing a band for each protein. Okay, now the steps in Western blotting is that first of all, a protein sample is subjected to electrophoresis on an SDS polyacrylamide gel. Then the electroblotting transfers the separated protein. So the proteins get separated, and by the process of electroblotting, it transfers the separated proteins from the gel to the surface of nitrocellulose membrane. Then the blot will get incubated with a generic protein. Generic protein can be, here we can see milk proteins or BSA. BSA means bovine serum albumin, or milk protein can be used as a generic protein. So the blot is incubated with either of these protein, which binds to any remaining sticky places on the nitrocellulose. An antibody that is specific for the protein of interest is added. So it can be taken as antibody 1, AB1, because we are again adding another antibody. So instead of getting confused, we can name it as antibody 1. So AB1, for the which is specific for the protein of interest, is added to the nitrocellulose sheets, and it reacts with the antigen. So only the band containing the protein of interest binds the antibody, forming a layer of antibody molecules. Next step, after washing for the removal of non-specifically bound AB1, the second antibody, another antibody that is AB2, is added, which specifically recognizes the Fc domain of the primary antibody and binds it. AB2, that is a secondary antibody. Second antibody is radioactively labeled, or is covalently linked to a reporter enzyme, which allows to visualize the protein in AB1 AB2 complex. So the applications of this Western blotting is that it's a confirmatory HIV test. It's confirmatory for the HIV test. It is used as a definitive test for bovine spongiform encephalopathy, BSC. BSC is also known as mad cow disease. It's actually a brain disorder in adult cattle that may spread to humans as well through diseased meat. Then another test application is that like some forms of Lyme disease testing employ Western blotting. Lyme disease, it's also caused by a bacteria that is Borrelia bacteria, which are spread by ticks. And Western blot test is a definitive diagnosis for Lyme disease. Another important applications is not written here is an evaluation of Western blot's ability to detect antibodies against Francisella tularensis, it's another bacterium, revealed that its sensitivity is almost 100% and specificity is 99.6%. So why this bacteria is that it is a bacteria that causes a disease called tularemia. Tularemia is a rare infectious disease. It's also known as rabbit fever or deer fly fever. It actually attacks skin, eyes, lymph nodes, and nodes or legs. So it's as I already told, it's caused by the bacterium Francisella tularensis. So an evaluation, so the Western blot ability to detect antibodies against this bacteria, its sensitivity is very high and specificity is also very high. It's nearly 100. And so it's another application of this Western blot. So that was my short video on the two blotting techniques. That was Western blot and Northern blot. Hope you get some points on that. So that's all. Thank you. Thank you so much.